I'm here with CanMed 2020 Cultivation Keynote Presenter, Seth Crawford from Oregon CBD. Seth, thanks for joining us.
Thanks for having me.
We wanted to bring Seth back to talk a little bit more about the process they use at Oregon CBD to create field-ready hemp varieties. He was recently a guest on the CanMed Coffee Talk podcast. Definitely check that out at canmedevents.com/coffee talk. And we were getting into the process, and Seth keenly pointed out that it was a little difficult to really explain things in such an audio medium. So he put together a nice video for us that really explains the process, and particularly a project they're working on at Oregon CBD, their varin projects. So through the magic of editing, we're going to put that video in right now, allow you guys to take a look at it, and I'm going to follow up with some questions. So without any further ado, here you go.
This is the slide that Ben referenced from CanMed 2018. It's a little bit infamous. This is an approximation of how at Oregon CBD we develop multiple new parental lines and then perform outcrosses for field trials before releasing a new variety to the public. And what you're seeing here encapsulates roughly 3 years worth of work to get to the point where these new F1s are released. It's important to note that any of our new public varieties require two distinct inbred parent lines to be created. Since 2017, all of our commercial F1s have been hybrids of a photoperiod plant and day-neutral plants. The resulting F1s are highly uniform, exhibit superior heterosis, and importantly, matured about the same time anywhere in the United States due to a change in light sensitivity brought about by the specific hybridization process we use. It also means that any competitors who attempt to inbreed our F1s will end up with 25% of the F2s auto-flowering, which, as you can imagine, is an undesirable situation for farmers. So our breeding process produces great F1s for farmers and essentially copy protection against competitors. The question is, how do we develop these parental lines in the first place? And a simplified answer is through targeted inbreeding with specific goals. The example that I'm going to talk about today is from the last 3 years of a very large project we've been working on. We call it our varinization program. The P1s and P2 used are themselves the products of this inbreeding process. It's worth noting that advanced breeding programs with diverse germplasm become circular. You lock in certain traits via inbreeding over the course of a couple years and then introduce new traits as they are discovered and begin the process anew. The varin program exemplifies this. We have three particular goals. One, as always, vigorous F1s to farmers. Two, creating varin versions of our entire catalog. And three, creating 100% compliant finished flowers that can meet the stringent USDA enforced 0.3 total THC. So we pollinated all of our production mothers with a highly inbred varin-rich P2. Then we grow out the resulting seed and test, test, test. The saying is, clone the best, kill the rest. In this program, the top 5 varin content F1s from each crossing combination were kept, cloned, and self-pollinated. As you're looking at this chart, these individuals are the peak on the far right. So when we say isolate and pollinate, we're taking clones of the same plant using STS to reverse one of the plants and then using that pollen to pollinate a copy of itself. We collect the seeds and repeat the process in the next generation. We're also simultaneously taking 3 of these plants and flowering them out in our research and development greenhouses for additional chemotype and phenotype testing. In the F1s, we ended up keeping 4.3% of the population and killed 95.7%. What was really interesting about this is there are lineage-specific patterns that emerge, and we've discovered that they're linked to CBDA and THCA synthase SNPs. So the genes responsible for making these compounds have varying catalytic ability of converting the precursor varin compounds. So the seed from the winners in the F1s were started, and the process is repeated. Testing, 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 and then cloning the best and continuing that self-pollination process. Now in the F2 population, we see maximum diversity. This is where selection becomes really, really critical. It's important in the F1, but it's a deal breaker if you get it wrong in the F2. This can be represented by some very basic statistics. You think about this in terms of mean and standard deviation. So in the F1 population, the mean proportion of varins as a percentage of the total cannabinoids was 11.4%, with a standard deviation of 3.2%. This changed to a mean of 14.6% in the F2, but the standard deviation increases by fourfold to 16.6%. You can also see this in the minimum and maximum values in these populations, with the really standout candidates in the F2 attaining up to 61% total varin content from the total cannabinoid fraction. So we select the best, again, kill the rest, and continue on. So the keepers in the F2 generation were approximately 1.3% of the total population, which means that 98.7% of the plants tested were rejected. So we continue this process until the population ends up looking like this. Visually, we can chart the improvement and see how progress is made by generation. When you're selecting the very best and the very highest content plants in terms of the varin proportion of the total cannabinoid fraction, you can make significant improvement between generations. So now we have stabilized plants that could end up as our new production mothers to make F1 seed. But first, we have to identify which crossing combination produces the most outstanding progeny. Just because mom is great doesn't mean the kids will be. So by selecting the best performers in outdoor field trials, real life circumstances, we can ensure farmer success when they use the seed we produce as F1s. The moms who produce the best offspring are then replicated in mass, and we start making seed. This is not a complicated process, but it does take time, 3 years on average, and it requires a lot of space, labor, testing, and considerable art in the final selection, but the outcome is always worthwhile. So it's been a long road, but over the last 3 years, our team has persevered, and we're really proud to say that we can offer fully compliant varieties to farmers in 2021 that have previously unavailable cannabinoids, CBDV, CBGV, and CBCV, as well as some additional traits not discussed today that are going to end up keeping farmers successful in a really meaningful way and represent a fairly revolutionary change to the way that we farm cannabis. It's basic breeding with modern science, helping to make farmers successful.
Okay, so the first thing that that really caught my eye and that I didn't realize when I was watching the video was that I didn't realize that all the varieties that you produce are crossed between one traditional photoperiod plant and a day neutral, which is also known as an autoflower variety. I didn't realize that you guys had done that, and I was wondering if you could explain a bit more the reasoning behind that.
Yeah, that's a great question. When you're trying to make an F1, the goal is heterosis, and it's the fancy term for hybrid vigor. To be able to achieve that, you have to have two parents that are very distinct genetically to maximize your chances to create that vigorousness. In the cannabis world, there's no plants that are further distinct than highly inbred photoperiod sensitive plants and highly inbred day neutral plants. They're very, very different. And we're actually in the process of being able to quantify that difference through whole genome sequencing at this point, with reference genomes developed for our day neutral varieties and for our photoperiod varieties. They're just incredibly distinct. And we settled on that technique because of the distance between those two parent populations.
So when you're doing a project like trying to breed for plants that produce the varin versions of the cannabinoids, well, first of all, for people who aren't familiar with that, can you explain a bit more of what that is?
Yeah, in traditional, quote unquote, traditional cannabis plants, we call it the wild type. This is the predominant version. The plant produces pentyl cannabinoids, which is basically just 5 carbon chain sides. The varins that we're describing are propyl, which is a 3 carbon side. All it really is just a slightly different version of the precursor chemicals, divarinic acid in this case, that gets synthesized by what amounts to the CBGA synthase. It's the GOT synthase within the cannabis plant, producing either CBGA when it's fed pentyls or CBGVA when it's fed the propyls or these varins. The CBGVA or the CBGA then traverses to cannabinoid synthases, which are either THCA synthase, CBDA synthase, CBCA synthase, et cetera, where they can produce both of these compounds.
And so what is really, I mean, if I was a consumer, why would I kind of be steered more towards the varin version of a cannabinoid?
It's a really good question. And at this point, there's some pretty good in vitro studies of the efficacy of both CBDV and CBGV of doing some fairly interesting things in the body, THCV as well. The experience is, I think, more energizing compared to regular CBD or THC or CBG itself. They're just a unique experience. And what's really cool about it too is that, as a consumer, you can be, either smoking it or you can be consuming it through tinctures or, other forms of preparation that allow for the isolation of these different compounds. So you can actually figure out what they do and if they work for you.
Interesting. Okay. So now going back to, you have one day neutral and one photoperiod plant, and you're also trying to breed for these varin cannabinoids. So are you having to do the same process for each type?
Exactly. So when we say we have to have 2 independent lines, we have to have an independent photoperiod line and an independent day neutral line to be able to combine to make that F1 that goes into a farmer's hand, which in this case resulted in us having to outcross. We had 14 different varieties that ended up getting pollinated at the very beginning of this process to create the varins. So we're independently developing CBGV day neutral, a CBDV day neutral, and then all of these other photoperiod versions of those as well, so that we can recombine at the end of the 3-year process and make seeds for farmers.
Wow, that's interesting. And so you get into this point where you're having to clone the best, kill the rest, right? I like that. And so each time you're doing this round of selfing, so how many seeds are you actually getting out of the plants? How many are you planting? Like, how quickly does this sort of scale up?
It gets problematic when you start talking about the numbers. And if you're, if you keep 5 from one particular line that has the highest initial total varin content in that F1, you're looking at growing out 100 minimum in the F2. And what we've found is that the more you can actually grow out, the higher the total content you're going to be able to find in those F2s. Our best populations that we've assessed at this point, we're looking at about 300 individuals to identify then the top 5 candidates in the F2 pool to basically restart that whole process. Once we're to the F3 generation, that's the S2, then the traits are pretty well locked in, and we can focus more on identifying the highest total cannabinoid content that corresponds with the highest varin fraction. So in those cases, we're growing out anywhere between 1 to 200 plants to make the next selection, and then saving the top 5 basically from those and doing the process over again. So it's a tremendous amount of testing when it comes down to it. And what's kind of interesting too on the production side, what we're going to be looking at for this next year is we have to test each individual day neutral plant to ensure that we're continuing to have that super high varin content as we use them as a pollen donor to make our varieties.
Yeah, and you mentioned that there's a lot of testing going on. So obviously you're doing potency testing. Is there any other type of testing that's kind of involved in this process?
Through this project and in collaboration with Medicinal Genomics and Eurofins, working on this SNP chip assay that will be presented at CanMed as well, we are working on basically identifying the genetic markers that are associated with varin and total cannabinoid content, so that we can actually go back and when we do this the next time, or if we improve a variety in the same way, a new variety, we can hopefully accelerate the process using genetic markers. At this point, we're still investigating, trying to locate those. It's one of those chicken and egg things, which is false because we know that eggs came first because of dinosaurs, right? Everybody knows that. In this case, you have to have the phenotype data and you have to have the chemotype data to be able to get the genotype data that can be used for rapid screening. So you can't do good breeding without all three at this point.
So obviously then, well, maybe not obviously, but the goal then is to be able to kind of pop some seeds and screen the seedlings, and you won't have to grow the entire plant up, and then you can kind of accelerate the entire process.
Exactly. And we're doing that, obviously for a number of traits, a number of agronomically important traits, so that you can get to the point where instead of screening 200 plants, you can screen 2,000. And rather than taking 2 weeks to do it, you can do it in 2 days. It allows you to rapidly increase the ability to weed out plants that would become non-selected at the end of the day.
And I imagine it allows you to kind of create a bigger pool to draw from too.
Exactly.
You're less constricted by the footprint.
Yeah, exactly. Yeah, when we talk about some of the initial crosses that you make for an F1, you can do good selection even with small numbers if it's a trait that's carried over and it's important. 1 in 20 out of that F1 will be an outstanding plant. But if you can increase that proportion so that you're screening 2,000 and now you have got a bigger chunk to draw upon, then you can find true needle in the haystack sort of one-off individuals within these really, really large populations.
So it's a numbers game. The more numbers, probably the better off you're going to be.
Yeah, it's kind of like science. The bigger the population size, the better the information, and the better the outcome in terms of the explanatory power. Excellent.
Any other previews you can tell us for the presentation that you're going to be giving at CanMed 2020 this year?
Some interesting information about whole genome sequences. We've been working on that in-house and collaborating with Oregon State University. We've got some fantastic better than reference quality whole genome sequences put together in the same vein that Kevin and folks at Medicinal Genomics have been working. We're using PacBio's SeqWell2 sequencer to make these things happen in-house. We have cannabinoid free, we have varin-rich plants, we have auto-flowering plants, and some other really, really interesting combinations that we have this whole genome data for. Something else that will be coming out, and this is kind of like the big drop for the 2021 season outside of having these fully compliant varieties, is being able to have seedless varieties. So we are introducing varieties that really can't be pollinated. And there's some really other cool attributes that go along with that particular trait that make the plants just basically super plants compared to what we've seen before in the cannabis world, which is beyond exciting. Like we're seeing the maximum expression of cannabis diversity, and not just that, we're seeing the maximum expression of humanity's ability to understand what's going on inside of the cannabis plant at this moment in time. So really exciting stuff.
So it's really interesting about all the different whole genomes that you've put together. So is the idea to kind of put together a pan genome that can really be used as sort of the ultimate reference?
That's exactly it. Yeah, and what we've discovered is if you're not doing de novo sequencing at this point, especially with cannabis, it's such a highly repetitive genome. If you're not doing that de novo sequencing, then you don't know. It should be a great tagline for PacBio. It's a wonderful piece of equipment, but we're opening up stuff that we couldn't see with previous short read sequencing technologies. And what we're finding is that those reference assemblies that are available, like the CS10 and now the wild Chinese cannabis plant that was recently uploaded, they're just not as complete. And the individuals that represent cannabis right now as a whole genome are not necessarily representative of the diversity that's in the population. So this pan-genome approach, I think, is exactly the direction that cannabis research needs to go. And the more genomes that we can pull in and be able to make public and aggregate while annotating with chemical and phenotype data, the more powerful this tool is going to become for humanity.
Excellent. Well, thanks again for the time, Seth. And
dude, no problem.
We'll be seeing you soon out there in Pasadena.
Sounds good. Thank you.
Thanks a lot.